SBOL, which stands for Synthetic Biology Open Language, has standard visual symbols which represent basic parts that help companies share synthetic biology systems. It is constantly updated to accommodate the development of synthetic biology. iGEM defines a basic part as a functional unit of DNA that cannot be divided into smaller components. Two or more basic parts join together to form a composite part, and if it serves a function in the cell, it is called a device. We will cover the most common parts in a DNA sequence: promoter, operator, CDS, RBS, and terminator. A promoter is where transcription of the DNA to mRNA first takes place. RNA polymerase binds at the promoter site, which is upstream to the transcription gene, and unwinds the double helix structure of the DNA. The transcription rate of the gene is controlled by the strength of the promoter, which is in turn determined by its gene sequence. Transcription is also regulated by activators and repressors. For instance, in a high tryptophan environment, tryptophan acts as a co-repressor to activate the TRP repressor. The active repressor binds to the operator on the TRP operon and prevents the transcription of enzymes needed for tryptophan synthesis because there is already enough of them. We'll talk more about this while discussing coding sequences, but let's return to promoters. There are two types: constitutive and inducible promoters. We assume that constitutive promoters don't have any input, as the RNA polymerase binds at a fixed frequency to the promoter, so that mRNA and the encoded protein is constantly produced, while inducible promoters do require inputs, as the RNA polymerase will only bind under certain conditions. CDS is the protein-coding DNA sequence that is turned into mRNA through transcription. The mRNA is divided into codons, or 3 nucleotides, which encodes for an amino acid in the polypeptide chain. The sequence usually begins with a start codon, which is usually AUG, and ends with a stop codon, which is usually UAA, UGA, and UAG. Examples of CDSs include activators, repressors, and reporters. Activators attract RNA polymerase while repressors prevent it from binding. Both are located near the promoters and affect the rate of transcription. The effects of both activators and repressors can be seen in the lac operon, which is used to create enzymes that break down lactose. When there is no lactose present, there is nothing to break down, so the translation of these enzymes is stopped by the lac repressor protein binding to the operator. However, when lactose is present, its isomer allolactose acts as an inducer to the repressor and inhibits its action, allowing transcription and translation to occur. The activator in the lac operon is CAP, which is supported by CAMP to allow transcription. When glucose levels are low, lactose needs to be broken down into a simpler form. Less glucose increases the levels of CAMP, which in turn allows CAP to bind to the CAP site and allow transcription to occur. Lastly, reporters output something that makes you track the processes in a genetic circuit. One example of a reporter is fluorescent proteins, or GFP. The ribosome binding site, or RBS, is the RNA sequence in the transcripted mRNA. Ribosomes bind to the RBS, and the translation of mRNA to protein takes place. It is usually about 6 nucleotides upstream of the protein coding sequence, which usually begins with the start codon, AUG. The gap between the RBS and start codon is important because it allows for optimal translation to occur. A strong RBS and promoter can work together to increase the transcription and translation rate. This can be tested using the fluorescent levels of GFP. Terminators are located at the end of the gene to stop transcription. E. coli terminators are row independent, which means that they have a stem loop that stops RNA polymerase and an A tail that separates the RNA from the DNA and RNA polymerase.